What a delight to be here. Thank you all very much for having me. I'm looking forward to it, talking with you today. And that was great. I've never been accused of rescuing our field before, but I feel fantastic about that. So I'm going to talk today about sort of part of my work. So we're not going to talk about Liberia today, but I'm going to talk about kind of the mechanistic work, the ideas of understanding kind of the pathways, the ways that experiences can change your body and how that leads to differences long-term in your life, as opposed to other things that can be talking about. So we're going to focus on pathways today. And now I can't advance my slides. So it's like such a bummer. Oh, cool. Okay. I'm going to talk about childhood adversity today. I want to start by kind of setting the scene so we can all be on the same page with our definitions and how we're understanding things. Adversity exposure is, I conceptualize that as an experience that a kid can have, which is really different than the expected environment that kid would be likely to be born into. And it requires significant adaptation on the part of a child. So you can think of things like maltreatment would certainly be adversity exposures, but also things like profound poverty might be conceptualized as an adversity exposure. Even if you're very broad, you can think of things like parental divorce, where you're sort of have an abrupt change in your caregiving as an exposure to adversity. And if you take that kind of a broad conceptualization of the term adversity and you include things like divorce and poverty, then 50% of children have experienced an adversity by the time they reach the age of majority in the United States. So it's a very common exposure. I think often we think of adversity, we think of really profound and very terrible things. We think surely that's not happening to very many kids. Some kids are experiencing profound and terrible things, but most kids are, almost most kids are experiencing, half of kids, some form of adversity during the time they're growing up. There's been a lot of work on this, but adversity exposure also increased during the pandemic. And we had a chance to take a group of kids who we'd actually been sampling before the pandemic and follow them weekly through the pandemic. And we wrote a couple of papers where we followed them actually for almost a year and a half. So a really long period of time. This is from just the first 7 weeks of after lockdown. And what we observed is that that big disruption in the family environment, that change in kind of the resources that families had in their routine and their capacity to kind of cope with everyday life, the abruptness of the lockdown led to a big spike in exposure to physical assault in the weeks right after the lockdown. So pre-pandemic, the kids in the sample, their parents reported 8 exposures to physical assault in the whole last year on average. And then within 1 week of lockdown, 76% of them had experienced a violent event with adult, and 67% had observed a violent event between adults. So a really profound increase. Really importantly, that decreased rapidly over the first 6 weeks. So as families kind of found their footing, they were able to find systems of care to support themselves and their kids, then you saw those experiences of family violence decrease. And I bring this up both because I want to sort of point out that recent events have been important, but also to sort of highlight the importance of context. It's not that there are just some families where this is always going to happen and some families where this is not going to happen, but actually something like a sudden disruption in schedule and in resources can actually increase risk for kids. So we can just know by this evidence alone, the kinds of context, the kinds of resources that families have are going to change the degree of likelihood that kids will be exposed to violence in their homes. We also know from just an enormous amount of work that adversity increases risk for psychopathology. So there have been lots of long-term studies looking at associations between adversity exposure and psychopathology. One of the interesting facets of this is that you can observe in population representative samples that adversity increases the likelihood that you're going to have a mental health problem as a child. But independent of that, it also increases the likelihood that you're going to have a mental health problem as an adolescent, and independent of that, the likelihood that you'll have a mental health problem as an adult. So it's this really long reach of exposure of early adversity. It seems to increase your risk later, even long after you're potentially out of those experiences. My work is, I'm having such a fascinating time, you guys. I never have technical difficulties, but I think this is going to be with us for the whole, I don't know why, but it'll be with us this whole time. So say hello to your little yellow friend. The question that I am interested in, as we talked about, is the mechanisms through which that long reach of adversity happens. And there are many potential mechanisms, right? If exposure to adversity can do all there are all kinds of different ways that might make it more likely for you to have a mental health problem later. There can be mechanisms going through your school experiences, mechanisms going through your friend experiences, mechanisms about just your relationship with your parents long-term that might yield this increase in psychopathology later. I'm interested in a very specific mechanism, and the reason why, what I'm interested in is this idea of biologic embedding, this idea that social environments that you experience as a child become embedded in your biology. They change the way your body and your brain function, and that is one of the pathways of risk for mental health problems later. And I'm interested in that very specific idea because this idea has a couple of things that are very fascinating about it for me. First of all, it dovetails really well with something we know a lot about from developmental neuroscience, which is this idea of plasticity. The idea that the developing body and brain are plastic, they're available for the experience to shape them. They're designed for that. The developing brain is intended to receive experience in order to help it develop better into a more competent brain for the environment in which it's about to enter. So it's very likely and almost necessary that these kinds of experiences would shape brain development through those typical developmental plasticity processes. The other thing that's really interesting about this hypothesis is that it explains this long reach. If you think about the body being changed, then you can imagine how that would show up later as risk for psychopathology, even if it didn't show up earlier as risk for psychopathology. So it's sort of the two things that I think this is a really compelling hypothesis. And there's been pretty good data showing that in general, adversity experienced during childhood has causal relationships with behavioral outcomes later. There's actually very little data showing that there's this evidence for biologic embedding changes in the body. And so what I'm going to show you first are some direct evidence of early biologic embedding. So I'm going to talk about that Bucharest Early Intervention Project as kind of causal evidence for the possibility of biologic embedding. Then I'm going to show some association studies talking about the different kinds of experiences that I think shape the brain and body. And then finally, I'm going to talk about contextualizing risk, how we should think about risk occurring in our daily lives here in the United States. All right, so first, is there causal evidence for biologic embedding? There are approximately 1 million studies of rodents showing that if you separate the rodent dam from the pups for between 3 hours to 24 hours, during puphood, that you see these really long-term changes in the pups later when they grow up. So they have an increased stress reactive response, their immune systems function differently, their gut microbiome function is different. There are changes in their neurochemistry. There's this really profound effect on pups. And these separations last between 0.3 and 3% of puphood. So it's a substantial percent of the entire time that they're pups. I bring this up because super interestingly, I wrote an op-ed about this a few years ago. Really interestingly, this is also the duration of time that children were separated from their parents at the border in 2019. It's the same duration. The range was a few months to a few years, and that same duration sort of, so I kind of argued in that op-ed that we might expect a similar effect that could happen to those kids as they were separated. There's been this other instance of random assignment to maternal care and out of maternal, or no random assignment out of maternal care, to be clear. We never did that, but there was random assignment to maternal care or to caregivers in a family in this Bucharest Early Intervention Project. And like Martha was describing, the utility of this project is that it's got this ability to make causal inferences. I'm going to tell you a little bit about this project to kind of set up what this is. So in Romania, this started in the year 2000. In the 1990s, there was a dictator named Ceausescu, who was a communist dictator. And he believed that what would help Romanians is if they had more children. So there's sort of a overarching finding in the world that if you have a more fecund populace, then you have a more productive economic environment. And it's kind of a classic causation correlation question. So all people who are in intro psych classes learn that things that are correlated are not necessarily causal. And in this situation, it's probably the case that if you have like a robust, happy environment, you have more kids, and then you have a more robust, happy environment in your country. But Ceausescu was like, let's just backwards engineer this. Everybody needs to have children, and then as a result, we'll have a more robust environment. So what he did was he carefully monitored the menstruation cycles of women, and he eliminated the capacity for them to find birth control, and he eliminated any control over their ability to decide when they would have kids and how many. And in fact, they had a lot of children. That did happen. And the consequence of that was that there was an enormous increase in poverty in Romania. It did not have the effect he imagined. It had quite the opposite effect. And as a consequence, families were unable to care for all the kids they had, and they established a system of state care to kind of deal with the situation. And the state care were institutions. And the idea was that these institutions would be places where you could bring your baby if you were not able to take care of it, and then they would take care of it in the institution. There were some terrible institutions, but most of the institutions were clean. They were staffed by people who intended to help the kids and wanted to, but they had this attribute. There was 12 babies and 1 caregiver, and that caregiver to child ratio would be not acceptable in any current place in America. We wouldn't, no one would ever have it be a reasonable way to take care of kids. By law, you have to have better child caregiver ratios than that. And but in addition, there was rotating shifts, so it wouldn't necessarily be the same caregiver every time that the baby saw. They would see them for a few hours and they would change. Sometimes they would move across institutions. So there wasn't any way to kind of establish a formal attachment bond with the caregiver across time. And those attributes of the institution had these very, very profound effects on the kids' development. And Chuck Nelson, Nathan Fox, and Charlie Zena started this in the year 2000 in order to study it. And they were specifically interested in this idea that maybe this lack of species expected environment, that the environment you should expect to be born in as a baby is some kind of familial care. That's how we're kind of all have been evolved to function. And that was not available to them. So they took 136 kids. At this time in Romania, there was no foster care. That was the established way, that the sort of like culturally normative way to take care of kids, if you can take care of them, was to put them in the institution. And so they established a form of foster care there. It was a kind of like the Cadillac of foster care, better than any foster care you would currently find in America. It had an on-call nurse who was available to talk with you 24 hours a day. There was home visitation by staff daily and then weekly, and then they were kind of consistently on call, and they would have regular times of follow-up for the first 5 years of the kids' lives. So until the kids were 5, they were in this really excellent foster care environment. What they did was they found 136 kids in Romanian institutions in Bucharest. They took half of them and they randomly assigned them to this foster care. They just picked the names out of the hat and they put them into this foster care intervention. The other half were in care as usual. They remained in the institutions. Now, Chuck, Charlie, and Nathan believed very firmly that being in the institutions was not a good form of care for these kids, but there actually wasn't a study of it. This is the only random assignment to foster care out of an institution that exists in the world, and it's probably the only one that will ever exist, because the results were so dramatic that at this point, they actually started a different kind of randomization out of an institution study. They had to randomize all the kids they could out of the institution and have sort of different forms of care for them, because it was just too profoundly obvious that this was a bad idea in terms of how to take care of kids. Now, I worked with Chuck on my postdoc, and right before I started, they had an MRI when the kids were about 9 years old, and then they did another MRI. And I've been working now, I'm still working on this project, and I've been sort of doing the MRI on the project since then. So they had another one at 16 years, and then we did it again at 21 years. I'm going to talk today about the 16-year data. So I said there was this profound effect on kids. If you just compare kids who were randomly assigned to foster care compared to kids who were not, what you see is that they have better mental health. They have a less blunted stress response. Their stress physiology functions more normally. They have improved IQ and cognition. So they actually have a higher IQ if they were randomly assigned to the family care versus foster care, versus institutional care, really importantly, institutional care. And they have better learning from reward. So they're better able to adjust their behavior to sort of like the possibility of getting reward. So really, like, that's a very basic learning mechanism. So really basic learning mechanisms up to really complex behavior is improved by this experience. I said we had MRI data collection on them. We have it on 115 kids at the age 16, and at age 9, we had a smaller sample of 64 kids. So the first thing at age 16 that we looked at is just to ask, is there a causal association between being randomized to foster care and your neural structure later? And we found there was. This is just these differences between these green and blue differences are 2 parts of the brain. where there was actually thinner cortex for the kids who had randomly been assigned to foster care compared to the kids who stayed in the institution. The structure of their brain is actually different. And what's interesting to me is that this part of the brain here is in the prefrontal cortex on the lateral on the outside, and the other part is the anterior cingulate cortex in the prefrontal cortex in the middle of the brain. And that's actually not where I expected to find these differences. So I had seen so many differences in behavior that I thought, yeah, we'll probably see some difference in the brain. I was hopeful, jumping into that data, being real confident. And we did see differences in the brain, but I thought we would see them in other parts of the brain. And that's because I had the idea that the prefrontal cortex, we know, develops for a really long time. It starts in infancy, but it continues all the way until you're about 25 years old. And I thought, with all of that long trajectory of brain development, of all the parts of the brain to be different, surely not this one, when the randomization is happening in infancy before the kids are 3 years of age. But here we are looking at it. We asked a couple of other questions. The thing I wanted to know is, what does this look like developmentally? Are we seeing just this difference at age 16, or is there a difference? Does this change how the brain actually develops? And it turns out it does. So if you look at the whole brain and just ask which parts of the brain are developing differently, you see all these areas in green and blue are areas that are developing differently in the kids who were randomly assigned to foster care versus those in care as usual. And the same thing's happening in all of these regions. That is for kids who are randomized to foster care. You see this thinning of cortex from 9 to 16 years. That's totally normal. That's what should be happening in cortex across that period of time. We should all have thinner cortex when we get older than we do in the beginning of our lives. That shows that the brain is developing typically. For the kids who have been randomized to care as usual, there was no change in their cortex across this period. So there is sort of a relatively flatter trajectory of change for those kids. And this was true everywhere. We found no evidence of the opposite findings. There's no situations where the opposite thing was going on. There's only this. We wanted to find out whether or not these differences in cortical structure were related to the behavioral outcomes that I described earlier. So we looked at we just took these two regions and we just looked at two different potential findings, mental health and IQ. That was because those are sort of the two places we've consistently seen differences in these kids across time. We found that thickness in the inferior frontal gyrus, this area on the lateral part of the prefrontal cortex, was correlated with IQ. So the thicker that region, the less normative, the lower IQ, and vice versa. The thinness led to higher IQ. Symptoms of psychopathology were associated specifically of externalizing psychopathology, so kind of rule-breaking behavior, were associated with anterior cortex thickness. So this is just a correlation. We are actually not controlling for group in these analyses, but it does show that these sorts of thickness might mean something in terms of how behavior is working. The final thing that I have done recently with my student Lucy Lurie is to ask whether or not, what it was about the environment that did this. So I think I certainly thought, obviously it's the caregiving environment. That's the thing that we changed, right? We did this really dramatic shift from institutional care into caregiving. There are actually lots of things that have changed as a result of that. So it could be that dramatic change led to big differences in the stress response, which we know it does. And then the stress response is the reason why we're seeing differences in the brain. Or that big difference is actually just a proxy for something like nutrition. And actually they're just overall body growth or their head size are going to actually account for the difference in this. We tested those things. We tested a bunch of things. None of them account for the difference. The thing that accounts for the reason why being randomized to foster care makes your brain development look more typical is the degree to which, when someone observed your caregiver working with you when you were between 30 and 42 months of age, the degree of sort of sensitivity and typicalness of your caregiving environment, the degree to which that caregiver scaffolded your cognitive development and also sort of interacted in a sensitive way with you as a kid. That's the thing that matters. It is actually the caregiving environment has this indirect effect that explains the association between randomization to foster care and cortical thickness. Sorry, here's the data values and the indirect effect. So as a result of this data, I feel like I can say with confidence that there are some examples in which we have evidence, which is causal, that early environments shape long-term brain development, not just the actual structure, but also the process of development in the brain. But as I described, I was puzzled about where this change was. I thought it would be in parts of the brain that developed earlier. So I was expecting to see it in like subcortical regions, which we know develop earlier, maybe in the back of the brain, which develops earlier than the front of the brain. And I found that sort of surprising. So I did a little study when I was at, oops, I had more explanation. So this is just a picture of longitudinally cortical thinning. I described that as normal earlier. I said your brain should start out thick and then it should get thin across time. This is an example of that. If you look right here, you can see the part of the brain that takes the longest to thin is the prefrontal cortex. That's that long trajectory of thinning. And so when I think about this data, I think we shouldn't see differences from early experiences in the prefrontal cortex. That doesn't really make any sense. But there is some other data that does make more sense. This is data from postmortem studies. So these are studies of people who have died for various reasons, typically. And you can see, if you just take their brains and slice them up and just count the number of neurons, there's this thing that happens all over the brain, which is a rapid increase in neurons, proliferation of neurons, and then a decrease in neurons that happens across early childhood and continues into adulthood. This is about 1 year of age, and somehow, some point here in the 1 year of age period is where you begin to see this decrease in neurons. And that's a slice of the brain from the prefrontal cortex. So, and I think I just said a bunch of stuff about increasing neurons and decreasing neurons, and I'm really sorry about that. For any neuroscientists in the crowd, I didn't mean neurons. I meant synapses. So I apologize for the neurons and the decrease in neurons. Switching between my public and professional talks there, apologies. So yes, increase in synapses, proliferation of synapses, pruning of synapses. This is an example of counting dendrites, so counting synapses. And this happens, we know from animal work and from postmortem studies, this happens all over the brain. And so that actually does happen in the prefrontal cortex. It looks like at that same kind of rapid trajectory, the way that it happens in the back of the brain. So I thought maybe this really early change in cortical thickness might be one reason why you'd see this long-term effect in the prefrontal cortex. And that to me has a lot of really interesting downstream implications. We know a lot about this process of proliferation and pruning in places of the brain that, like the visual cortex, where we understand what kind of inputs are needed to shape it. We understand what those inputs do to the structure of the visual cortex across time. There's sort of a really well understood process of developmental plasticity that is characterized by this rapid increase and decrease in synaptic connections. And I happen to have a data set that I worked with Wei Li Lin. I collaborated with him and got this data set from him that he had collected, where we had really densely sampled imaging in the early part of life. So we have it from, we had imaging sampled during this first year every 3 months. So right when they were born, 3 months, 6 months, 9 months, 12 months, 18 months, like really rapid acquisition of signal. So we could actually observe this rapid increase in cortical thickness and this decrease in cortical thickness that would be predicted if we were capturing something like proliferation and pruning of synapses. It may not be that. We don't know what it is. It's cortical thickness. But that's what we would think it would look like if we were capturing that process. And that's exactly what we saw. So the first thing we do is we try to figure out how to model this. This is like a difficult to model trajectory. So we did a good job of figuring out, like, what does the shape of this actually look like? We did it 3 different ways. We modeled it using a specific model designed to capture the shape. We also modeled it using a GAM model, and then we did it again using a piecewise HLM. So we did it many different ways. And every single time, this is just zooming right in on this period between 0 and 2 years of age. What we saw is this increase in thickness followed by this decrease in thickness with a peak around 12 months. 12 months. The next thing we did is we asked these same kids to come back in and we imaged them doing a working memory task, a task where they had to hold something in mind for a few seconds. It's a difficult thing to do. You have to hold something in mind for a few seconds, and I ask you tough questions and make you work hard at it. We did this 2-back task, so a 0-back and a 2-back condition where you're told 2 things or just find 1 thing. So a very difficult thing and easy thing. And we found that they activated parts of their brain that we would have expected, given what we were asking them to do in the prefrontal cortex. And we asked, does the degree to which this shape in cortical thickness, this increase, the peak it reaches, and the decrease, do any of those aspects of the change in cortical thickness predict the way that they use their brain to do that working memory task later, or to predict how good they are at it? And the answer is, it does. So really interestingly, the degree to which, how tall your peak is of cortical thickness, the degree to which you thicken, at 1 year of age predicts both how good you are at that working memory task and also how much you activate your brain when you're doing the task. And it happens in exactly the same part of the brain. The prefrontal cortex predicts the prefrontal cortex activation 9 years later. So it's across a really long period of time, and thickness changes and no other part of the brain do that same prediction. So we checked all the other lobes and none of those do it. And we controlled for multiple comparisons. So overall, I feel like this is pretty interesting evidence that I'm kind of intrigued as a scientist. I want to find out what else we can learn about how that prefrontal cortex is using that information, what signals it's listening to, what it's actually interested in the environment, and to know how it should shape itself for its future. Because not to anthropomorphize too much, but that is the goal of the brain, right? Is to get information to come in to figure out how to make the most efficient brain for the environment in which that person's going to live long term. And the idea that the prefrontal cortex is doing that in this really early period in infancy is predicted by everything we knew about animal work, but is still really exciting, I think, to be able to see. So I think it's really very interesting to see. Now, the rest of my talk is going to be about what the signals are I think the prefrontal cortex is paying attention to, and also sort of the brain in general. What is it looking at in the environment in order to learn how to be the most efficient brain for the rest of that child's life? And one of the places I'm going to look is in caregiving experiences, obviously, because that's the place I looked at the Bucharest Early Intervention Project, but I'm going to kind of define those experiences more broadly, the way that Martha was describing. Does anybody have questions right now? Okay. Okay, so here's the question. The question is how? How is the prefrontal cortex being shaped by early experiences? And my colleague Kate McLaughlin and I have this theory that it's there are different dimensions of experiences that kids have, and those dimensions of experience are going to shape their brain differently. There are lots of different ideas. So this is a figure sort of showing these different ideas, and I'll describe it briefly so you can get a sense of why this is different. And Martha set this up really nicely. So you could have the idea that each different kind of experience that happened to a kid, if you think of all the different bad things that can happen to kids, each one of them would have its own effect, right? If you're exposed to physical abuse, that would have one mechanism through which it shaped your long-term risk. If you're exposed to parental separation, that would be a different mechanism that would increase your risk. That's one way of thinking about things. One different mechanism for each kind of experience, really thinking about them all distinctly. The other way you can think about it is sort of a cumulative approach, which is what Martha was talking about, which is like 1 is bad, 2 is worse. By the time you get to 4, you're almost certainly going to have some kind of a bad outcome. And if the only thing you're interested in is general health outcomes, this is actually the way you're going to want to go. This is a great way of predicting general health. It is true that if one thing happens to you, you're likely to be robust to it on the level of the population. And if 4 things happen to you, it's more likely that you're going to have some kind of problem later. But we don't really know what problem or why it's happening. And it's certainly no way to look at developmental plasticity, because the brain isn't interested in like bad as a category, right? The brains are very specific. They want to know what exactly is happening and how to shape that particular part of the brain. The idea here with deprivation, we had this idea that perhaps we could think of there being dimensions, so that the experience that's happening to the kid could be categorized as something like deprivation, a lack of species expected inputs, the lack of typical care, a lack of stimulation in the environment, a lack of richness in the kinds of experiences that kids were having. So the brain didn't have as much input or as rich input as it would be expecting to have. Another dimension would be something like threat or harshness. So the idea here would be that it's the presence of learning experiences that are very salient and that have a lot of negative associations with them, or negative affects. So they're traumatic experiences. There's the risk that they themselves will be harmed, or their close other will be harmed. It's usually interpersonal violence, and it's usually sometimes in the home, but it could also be outside of the home. The idea is like if you experience interpersonal violence, any of those places is going to be an example of threat exposure. There's another dimension, unpredictability. I'm not going to talk about it today, but we're developing some theories about the mechanisms through which unpredictability will work. And I've got some interesting stuff happening in the lab. Sorry, I'm not going to tell you about it, but in the future. So there are mechanisms in this cumulative risk model. The mechanism is stress. So I think most of us have probably heard that stress is bad for your brain. And a lot of the ideas about why stress is bad for your brain and the fact that stress is bad for your brain come from this model. And this is true. Too much stress, toxic stress, is bad for your brain. And toxic stress also does shape the way that your sort of body functions long term. And it shapes the way that it increases your risk for psychopathology. But this is probably not the developmental mechanism that I'm thinking of when I'm thinking about why brains are different across time for kids. Stress is not an input that the brain is receiving. Stress is sort of the body's attempt to regulate itself in response to constant disruptions. That's not the kind of thing that your brain has evolved to like shape part of your cortex for. It's like a disruption, not a developmental input in the same way, I think. So while it's definitely bad for you, it's not the kind of developmental input that I thought we needed in order to be able to understand how things worked. So when we thought about these dimensions, we tried to think about what they would impact. So first we kind of divided up the different known exposures, the way people were thinking about them, into bins, deprivation and threat. And then we tried to think about, okay, if we have something like physical abuse or physical and sexual abuse, domestic violence, and community violence, what would that dimension look like? What kinds of things do we think it would impact it? We looked into the animal literature. The animal literature says if you expose rodents to threats, to interpersonal threats, either you do the foot shock, you give them a dam where you disrupt the resources the dam has access to, and so the dam functions in a way which is sometimes abusive, drags the pup, rough handles the pup. If you do that, the result is that the pups they acquire fear learning earlier. They're better able to detect in their environment when something bad is going to happen. Makes a lot of sense. You're like, that seems like a useful skill. If you're going to be born into an environment with a lot of violence, seems like a good idea to try and figure out how to predict that. They also have higher reactivity and it stays high, especially during puphood. So that was our hypothesis there. We thought it was going to disrupt the fear learning mechanisms in the brain. So the parts of the brain that do fear, which are broadly the amygdala and the ventral medial prefrontal cortex. And we thought that it was going to disrupt emotional reactivity in kids. On the other dimension, deprivation, we looked at this group of institutionalized kids who we've been working with for a long time, and we thought, like, what is it that this lack of stimulation is doing? We think what it's doing is it's not providing enough information of their brain that it needs to keep its rich dendritic arbor, that it needs to sort of be ready to solve complex problems later. And as a result, we think it might maybe over prune and not be able to do the same complex cognitive functions that are required later. So specifically, we thought maybe like really abstract things like language or executive function, that working memory, that ability to hold things in mind, maybe those will be disrupted as a result of this lack of rich inputs early on. And this is sort of a drawing out of our hypotheses here. So we thought that deprivation, which we would measure as things like neglect, but also cognitive stimulation, social stimulation, linguistic inputs in the home. And then threat, which we would measure as physical and sexual abuse, interpartner violence, and community violence, would impact different parts of the brain. It would either impact the executive function or cognitive control network, or it would impact the ventral medial prefrontal cortex, the amygdala, and the salience network. They would lead to these cognitive differences in language, executive function, and cognitive capacity versus fear learning and emotional reactivity. But both of those would lead to increased risk for psychopathology. So we're not hypothesizing you're going to get different disorders. We think all disorders your risk for disorders will be increased by both forms of risk, but they'll just travel different pathways to get there. So we set out to test those things, Kate and I, and we wanted to show, I want to say actually one thing about these, first of all. These are proximal inputs. So the thing I'm measuring is not whether or not parents want to help their kids, or whether or not they love their kids, or whether or not they are intending to be good parents to the kids. You think back to that stuff that I was talking about when I was talking about the COVID pandemic. The environments that people are in, that families are in, shape the way they're able to interact with their children. So this is just proximal inputs. If you have to work 3 jobs in order to make ends meet, you might not be able to provide the kind of sensitive care that you hope for your child. It might not be available to you because you're trying to make sure that they can eat. And as a result, they might have what we would then characterize as deprivation. But that doesn't indicate that you wish they didn't have deprivation. That just indicates what's actually happened to the child's brain. So this is really different than when people sometimes measure kind of like the warmth or intentionality of the parent. They're kind of like the way that they function when they're at their best. That's not what we're interested in. We're actually interested in like literally what is happening to the kid's brain as they're growing up. also the idea is because there's such proximal inputs, we're thinking about how these proximal inputs shape brain development. We're thinking of very proximal outcomes. I'm thinking of something kind of esoteric, your executive function, your ability to hold things in mind for a few minutes or solve problems, not something I think that will ultimately lead to differences in your educational opportunities and also in psychopathology. But I'm thinking of the pathway being like a very proximal thing that needs to be measured in the brain and through tasks and behavior. Okay. So we think these different dimensions are going to affect brain and behavior differently, and we think that they're going to be pathways to psychopathology. So we did a bunch of studies since then. We published this first in about 10 years ago, and we did many studies, and I'm going to tell you about a few of them. So first of all, one of the things I was excited to do when I got to Chapel Hill is, which there's like a theme here. I found some folks who already had data that was longitudinal, and they got them to let me analyze their data. And so what I was able to do is in a couple of studies, there was these two different studies. One's the Future of Family Studies, and the other one's this Child Development Project. They happen to have measured pretty well a bunch of experiences that we would call deprivation and threat. And this is work done by Adam Miller, who was then my postdoc and is now a professor, and Laura Macklin, who's at the NIH. So we were able to measure these. They had these well measured, these kids, when they were pretty young. This one, it was between 1 and 3 years of age. And then a few years later, when they were 5, they had verbal abilities measured well. In these two studies, they'd measured verbal abilities the same way. So that's very convenient for us. They'd also done this measures of deprivation and threat the same way, very convenient for us. And then years later, at year 15, they've measured their psychopathology, their symptoms of internalizing and externalizing psychopathology. So things like depression and anxiety versus their sort of like rule-breaking behavior, those two kinds of things. And we're able, through a statistical model called a path model, ask what is the path? How do you get from here, these exposures, all the way at year 15 to these outcomes? And the answer is that if you have threat exposure, it doesn't go through verbal abilities. It goes straight to psychopathology. There's no exposure here, which we would call emotional reactivity. There's no outcome, which is emotional reactivity or fear learning. You just go straight from threat, and you increase your risk for internalizing and externalizing psychopathology all those 12 years later, from years 1 to 3 through years 15. If you have deprivation exposure, really interestingly, it goes through verbal ability. So the path goes from deprivation through verbal ability to internalizing and externalizing psychopathology. You increase your risk for those kinds of forms of psychopathology, but you do it through the path of changing the verbal capacity of the kids as measured at year 5. So these two studies were our initial observation that what we thought was going to happen did happen. There was different paths for deprivation and threat. We're putting them in the same model. These are both modeled as latent variables as our psychopathology variables. Verbal ability is the Peabody Picture Vocabulary Test. So we had 2 examples of that, but so far we hadn't had any examples of these existing longitudinal studies where someone had done a nice job of measuring emotional reactivity. And then I met Kathy Proper, and I shared this student, Nicholas Morgito, who is fantastic and going to be looking for a job sometime soon, so get ready. And they had this Durham Child Health and Development study where they had 206 mom-kid dyads. They had measured deprivation. Deprivation and threat here are measured in an okay way. So it measured cognitive stimulation, which was great. And they measured it at 6 months, 12 months, 24 months, and 36 months. And then they measured threat. Threat is intimate partner violence. So we just had this one assessment of threat. It's not that good, but it was better than nothing. So we gave it a shot. And we had really nicely in this study. So this is when they're before the age of 3. This is when they're 5. And this is when they're like 8 or 9. So at 5, we have executive function measured beautifully, effortful control questionnaire measured beautifully, emotional reactivity measured beautifully, and questionnaires about emotional reactivity, which are really nice. And then at 84 months, when they're 8 or 9, we have their symptoms of psychopathology, and now we also have their ability to do well on tests of like math and reading. What we found is that socioeconomic disadvantage, this is kind of our sexual theory, is that socioeconomic disadvantage is leading to resulting in increases in deprivation and threat. So it's correlated with increases in deprivation and threat. Those are leading to differences in effortful control and emotional reactivity. And those are leading to differences in internalizing and externalizing symptoms. And the indirect effect from social disadvantage all the way to psychopathology was through threat and emotional reactivity. Interestingly, the link between socioeconomic disadvantage and achievement on math and reading tests was through deprivation and executive function as measured on those tasks. And then we test a bunch of opposite models because we were like, sure, this is what we thought was going to happen. And the model fits well, but like maybe something else is happening. So we'll switch it. We'll say like, do we still get a good statistical model if we ask if threat predicts executive function and deprivation predicts emotional reactivity? And the answer is no. We did all different kinds of inverted models. None of them fit. The best way to describe the data is the way in which we have, consistent with the theory, which is that deprivation is predicting executive function and threats predicting emotional reactivity. Okay. I'm going to tell you about the brain, and I'm going to tell you about the fun parts of the brain. So there've been a number of studies. I'm just going to tell you like the first one and the last one, and then I'm going to talk about something else for a minute. So, reminders of our theory, we think deprivation is going to shape the brain to lead to executive function. So far, I've just showed you deprivation, executive function, and language and psychopathology. So now I'm going to stick the brain in there. Okay. So first of all, this is just saying like, we've done a number of studies. We have a bunch of stuff in institutionalization, like I just showed you. We also have some studies where we've just measured directly or asked parents about the degree to which they're using cognitive stimulation with kids, whether or not they're neglecting the kids. We've asked kids whether or not they were neglected by their parents, and we've asked about specific experiences that parents have or haven't exhibited with them. And then we've measured differences in executive function and language. And in those studies, we control for things like the amount of threat the kid has experienced. So we ask like, regardless of how much threat, is there an association between neglect and lack of cognitive scaffolding and executive function and language? And consistently we observe there is. And there are two meta-analyses, which are like studies where you look at all those different findings together that confirm those observations. The first study that we did on this was a small study of just 51 adolescents. And we asked them again to do this working memory thing, to hold something in mind for a few seconds. And here we're asking them to hold in mind stars, so lots of stars or just a few stars. When you do that, you have to activate your cognitive control network, which your parietal cortex, this region here, is a part of. And what we found is that parental education, which this is our very first study, so it's a proxy for deprivation exposure, but also parent report of neglect, so the degree to which they report neglecting behaviors, both predict executive function in the kids. And parental education predicts this parietal cortex activation in the brain. And these predictions are even if you control for maltreatment. And we actually selected this sample to be a group of kids who had experienced maltreatment. So there's a fair amount of maltreatment in the sample. 38% of them reported a behavior that we would have considered maltreating during their childhood, a behavior, physical or sexual abuse. And this is their kids report. This is kids' report of neglect, and then the parent report of their education attainment. So we found this differential effect on that part of the cognitive control network, like we predicted. And then we went on to do a number of other studies where we replicated these results partially or in full. One of the studies that we did, and the thing that I'm most interested in, is getting back to that early childhood. So all this study and then the subsequent studies were all in adolescence, but I wasn't really interested in adolescence. I'm interested in this idea of early childhood. I wanted to know whether or not that embedding was happening early and then affecting long-term differences. And so I kept wanting to move our age ranges down as much as possible. So the first thing I did was a pilot study. And in that pilot study, we did this cognitive control task or executive function task. And we show that parental deprivation, controlling for threat, and age and gender and IQ predicts the degree to which you're able to do this this task where you have to stop yourself from doing something you don't want to do, and instead hold in mind this, use your working memory to hold in mind your future goal. That study was a pilot for what's been going on in my lab now for the last 5 years, which is a large study. We have 275 kids. We're finished enrollment now, so we're just analyzing data. And we recruited them in using indices of structural inequality. So we recruited in families who had at least one parent who had a minoritized racial or ethnic identity, who reported having less than a high school degree or low income, or who self-disclosed violence in the home. So all of those would get you into the study. And we were thinking, if we look at these indices of kind of structural inequality, we'll observe differences in exposure to adversity, and we did. So 62% of the sample reported using harsh physical punishment with their kids, and 22% report neglect. The kids are between the ages of 4 and 7. It's a pretty exposed sample, and it's often the first time anyone's asked the family about abuse and neglect. We ask both the parents and we also ask the kids. We do an interview that you can use with kids to ask them about it. I live in North Carolina. It's actually pretty common to use corporal punishment there. Parents are very open about their use of corporal punishment. They usually feel like it's the correct form of how to work with their kids, so they're not trying to hide that information from me often. They're trying to tell me that they're appropriately disciplining their kids when those kids need it, is how they it's understood. But it also is really a range. There's probably 10 cases where it was the first time that abuse or neglect had been observed with the family, and we had to report it to CPS. And there was a number of other cases where it had previously been reported and we didn't need to report it. So it's a pretty exposed sample. It was intentionally identified in that way, try and get kids there. It's also a long study. There's 3 visits. So they came in we went to their home, they came to the lab, then we did an imaging study with them. We measured deprivation and threat really deeply because that's the thing we're interested in. So we have lots of measures of it. We ask parents, the kids, and all kinds of things. And we do an observation with them. So really trying to get at that. Then we've also really richly measured executive function and emotion dysregulation. And then finally, we measure psychopathology with a structured interview. And we have a protocol paper out, and we're just analyzing the data from this now. So I'll show you some highlights. This is in 4 to 7-year-olds. We did, again, an executive function task. Again, this is one where they have to stop themselves from doing something they don't want to do and hold in mind their future goals. And if you ask them to do that, what you find is that, first of all, we see activation in the brain, which is what you would expect, which is great news. You see a bunch of parts of the brain that are usually active when you do this kind of task. That's good news. And then we specifically see the deprivation predicts activation in the cognitive control network where we thought it would. So you see less activation in those areas of the brain for the kids who have more deprivation exposure. What about the threat pathway? So same studies. First of all, I have to tell you, generally we've done lots of work. So physical, sexual, and emotional abuse, exposure to crime, community violence, those are our indices of threat. We see that they disrupt fear learning. They decrease automatic and explicit emotion regulation. They're associated with disrupted fear learning and decreased automatic emotion regulation. They're associated with disrupted amygdala activation and differences in amygdala, hippocampal, and prefrontal cortex volume and surface area. And there's a meta-analyses that confirm these findings. Across a bunch of studies, we've used this paradigm that I'm going to describe to you guys because this is kind of the fun part. I think it's fun. So we tell people to do something. These are adolescents. So we say, the next thing you see, I want you to just look at it naturally, experience it like you would experience it. Then we show them a picture. It's either an unpleasant picture or it's a picture of like a vase. And then we ask them, how do you feel about that picture? And then we do it again. Sometimes we say, just look at the picture and feel it naturally. And sometimes we say, when you see the next thing, what I want you to do is try and think about that picture as if it's far away from you, kind of decrease your reaction to it. It's not real. Remember that this is just a study and it's not a real thing. So try to sort of reappraise the picture. It's not really a bad thing. I can kind of decrease my reaction to it. This is a really common paradigm that's used a lot. We used it here, and what we saw is that kids who had experienced threat, this is the same study as I showed the cognitive control network. This is the first study that we ever ran. We see differences in their amygdala activation and also in their ventral medial prefrontal cortex activation. I'm not going to tell you about this. We replicated this a number of times. That's a replication that we did in 136 youth in North Carolina. Okay, I wanted to tell you this in little kids, and I didn't know how to do it because I thought that if I told little kids, next time you see a picture, think about it differently, they would be like, what are you talking about? And so I thought about how it is that little kids, what do we actually do with little kids? And I thought what I do with my 5-year-old is I say, sweetie, it feels really difficult right now, but it's actually fine. It's just going to be a little shot and then it will be all done. And it's okay. This is going to make your body healthier. That's why we have to do it. And then I'm hoping that my little 5-year-old's like, I'm going to have a healthy body, not someone's going to jab me with a needle. And as a result, then she'll feel better able to handle that situation. So what we did is we found a paradigm that folks had run before and we adapted it for the scanner, where we had parents actually say scaffolding information. The next set of things you see, they're hearing their parents' voice in the scanner. The next set of things you see, don't worry about them. They seem scary, but they're okay. And then we had a research assistant recorded telling an actual story. The next thing you see is actually a parent playing hide and seek with their kid. It's not a parent yelling at their kid, which is what you might think. And then we said, how do you feel about this? Thumbs up or thumbs down? They're little tiny humans, so you can't be like, scale of 1 to 10. When we do that, so we have pictures that are just look at this naturally, and they're pictures that look like this. We have pictures that say, this girl's in trouble. So we say some pictures where it's like, look naturally. When that happens, we do also tell them a story. We tell them a story that matches the picture. This girl's in trouble and her teacher is mad at her. Or we have them do stories where it says, this is like an example. It's a reappraisal. The reappraisal isn't matching the picture, but you tell them a different story. This parent's playing hide and seek. That's why they're looking at them in the closet. So you can sort of do these two different things. What we find is that kids report feeling better. If you give them a story that's like, don't worry, they're just playing hide and seek, then kids are like, I feel better about that picture. I don't feel as bad. I give it more thumbs up than I give it thumbs down. And they activate a network of regions in their brain, which are exactly what adults do when they're looking at the same thing. So this is like across a bunch of different studies of adults, and you can see these areas in the brain are activated in exactly the same way in little kids as they are in adults. So we felt like, so cool. We managed to create an emotion regulation task for use with kids. I had a friend who was an adult do it, and she was like, what is that gaslighting task that you did with me? And I thought, yeah, it's not useful for an adult, but maybe it's useful for a kid. So the next thing we wanted to ask is, what happens if your mom is telling you to get ready for what's going to happen next for somebody that you don't know? So this is my kid, this is me, and this is me saying, don't worry about it, Ollie, things are going to be fine. This is my colleague, Kristen Lindquist, and she can also say, don't worry about it, Ollie, things are going to be fine. And he's going to see either my picture or Kristen's picture and hear my voice or Kristen's voice. Who's doing it? The story voice is always not your mom. So it's just this quick manipulation before they see anything, letting them know to feel safe or not feel safe. And what we find is that there is a slight difference in the degree to which they activate their brain. They activate their brain slightly differently if their mom scaffolded them or the stranger, and they actually report feeling differentially better about these stories. So they're all reappraisal stories, but it's like, who told you to listen to the reappraisal story? It's in a very weird direction. It turns out that if somebody who's not your caregiver tells you to not pay attention to what happens next, you listen to them and you feel better the next time. But if your caregiver tells you that, you don't feel quite as much better. The degree of activation, it's the same regions that are used in reappraisal, but they're activated more when the caregiver has scaffolded them versus the family. So we're sort of still figuring out what to make of that. Okay. I'd like to do a little bit more, but I know we're late. I wasn't sure how this, can I do a little bit? I would really love like 5 more minutes. 5 more minutes. Is that okay, guys? Are you willing to do it? I appreciate you. There's cheese. So if you wait for 5 minutes, then there will be cheese for you. Okay. All right. So as I began my talk, I talked about the context in which people experience adversity. And I have disassociated generally in my work until recently those contexts from what I actually study. But social inequality is the context of adversity. That's how I sampled my participants in order to be able to attain a group who had a lot of adversity exposure. That absolutely patterns who gets adversity exposure in America. And I wanted to know more about that and study it in a more deep way to kind of recontextualize the adversity experiences that kids were having. So I had a study, this is a totally separate study, I haven't told you guys about it at all, don't worry about it. There's a study of 917 kids. They were first sampled between the ages of 2 and 5. We're following them up. This work is just with this early sample. It happens that this is a representative sample of kids in Durham. They went to all the well child visit places and just asked parents to participate. And they found a group of parents who are willing to participate who represent Durham County in North Carolina. So it's like a really good way of finding out what's generally happening in North Carolina to folks who are living there. There's a bunch of them. There's 917 of them. Because we knew where everybody was living, we were able to go and find out what their neighborhoods were like. And we did that using just administrative data. There's a bunch of different sources of this data. You can just ask, in this neighborhood, in this census tract, what are the educational opportunities like for kids? What are the health opportunities like for kids? What's the social economic environment like for kids? You can just go find it out. This is called the Child Opportunity Index. You just use the census tract. That's all we did. We then went to ask whether or not the impact of threat exposure, which we also knew for this sample, was on psychopathology risk, was different if you lived in a neighborhood with lots of opportunity who live in a neighborhood with not lots of opportunity. What we found is something that we thought we would find, which is that if you live in a neighborhood with very little opportunity, the association between threat exposure and psychopathology is much stronger. So this is showing how strong the association is. And you can see this line goes up, and it goes up on this side when you're in a low opportunity neighborhood. And the specific form of opportunity that mattered here was social and economic resources, kind of like the aggregate amount of money that's being made in that neighborhood. Now, that was what we expected to find, but we probed it a little bit further and we found some surprising things. So first of all, we found that overall opportunity also mattered: education, economics, health, but it really only mattered for kids whose parents identified them as white. So kids without racism exposure were able to benefit from overall economic opportunity, and kids with racism exposure were not able to benefit in the same way in their neighborhoods. And then we found that kids who are white also had a direct effect of education on their psychopathology risk. The more educational opportunity in their neighborhood, the lower their psychopathology. The kids with racism exposure, kids who were identified as Black, didn't have that same reaction to their opportunity. And this actually makes an enormous amount of sense if you find out what neighborhoods look like by white and Black participants in Durham, North Carolina. This is the plot of opportunity from 0 to 100 on the x-axis, and this is the density of participants who identify as Black and participants who identify as white. And as you can see, the way sociologists use index of opportunity, like the one that we use here, is they use it to talk about segregation, because segregation is what patterns people into neighborhoods. That's the reason why you have differences in opportunities across neighborhoods. And it's not on accident. It's very intentional. In Durham, there's a whole system of historic moments when decisions were made in order to change who got to live in which neighborhood, and then thus what opportunities kind of aggregated those neighborhoods. Now, I bring it up because I'm really excited about this, the future direction of research. I want to recontextualize the experiences of adversity. I want to think about how they interact with brain development. There's this really interesting new work that talks about how exposure to racism is related to some of the same pathways that we see in our threat exposure. So there's this really possibility of this being like synergistic and interactive at the level of the body. And I don't know what you guys think when you look at these plots, but what I think is that I can't not study racism and still understand what's going on in my participants. I have to do it. I would be a bad psychologist and I would be a bad neuroscientist if I didn't look at this. So no matter who says that I can't do it, I don't know how to actually understand what's going on with my participants without it. And that's why I hope I'll be able to continue doing it. So far, no grant has paid for this research. This was from the early part of the data, but maybe they will. My takeaway from that part of the talk is just we have to diversify our samples. I wouldn't have known that if I didn't have this representative sample of Durham. I could have read a bunch of sociology literature, but I wouldn't have been able to apply it to my data. We also have to embed context in our questions. Without looking at the context, we can't understand what these experiences really are. And if we don't study racism, we can't identify pathways of risk and resilience for all of the children because they're just so different for the different kids. All right, I want to thank my lovely lab and my funders and you guys. Thank you so much.